So you may hear random noises in the background. There's a baby at my base here. So I'm very excited to introduce people today because I think this is quite a good session and a topic that tends to be a little bit ignored in lots of fields, but autism in particular. And that's talking about epilepsy. So epilepsy is significantly more frequent in autistic people, and young children born that you'll hear from Charlotte, infants who are born very early, and infants who have early seizures are more likely to be autistic themselves. If you go to people who have treatment-resistant epilepsy, for instance, who may end up going for surgery to cure their epilepsy, up to 1 in 8 will be autistic people. So it is a really quite severe onus on autistic people. So I'm going to be introducing first Francisco Ferreira, and his father will be alongside, who's taken part in LEAP epilepsy. And in addition to the quite substantial amount of time they donated to us for that study, he has also agreed to talk to us a little bit about his experience with the study and his experience as an autistic person with epilepsy himself. After that, I will introduce you to Chirag Mehra, who's been running the LEAP epilepsy study here at King's, who's a pediatrician and also a very talented PhD student. And finally, I'll introduce you to Charlotte Tai. So where Chirag has been working with people between the ages of 6 and 30, which is kind of the primary LEAP cohort in AIMS trials. Charlotte has been working for years on separate studies looking at epilepsy in infancy and often genetic causes of epilepsy in infancy, which often co-occur with autism. Tuberous sclerosis being the first one, and she now runs a newer study looking at infantile spasms. Okay, so with that, Francisco and Jose, if you're happy to go ahead. The one thing I would say is that if people have questions, please, if you could keep them to the very end, but add them to the chat whenever you like. Okay, Francisco and Jose.
Go for it. One of the things I did enjoy.
Introduce yourself.
All right, yes. So, my name is Francisco Cancio. This is my dad, José. Yeah, José. Oh, José, Michael. And I've been off epilepsy for about a year.
A year
now. And so far it's been okay, I guess. It's weird not taking medication every other day. That would be doing. Very much last thing.
And just to introduce a bit more, in respect to Francisco, of course, his first case of epilepsy seizure was a 35 minutes episode at the age of 3.5. So just to give you a bit background in that respect. And then he had consequently a year after another one of 35 minutes and another one 2 years later of 20 odd minutes. And then the consequence of that, of course, was the start of medication at the age of 5, actually, because it took about 18 months to start the program in that respect, which then he had very seldom cases of manifesting physically the epilepsy. He had 2 cases of epilepsy, which were overnight, more or less non-physical, non-manifestation, and he would actually woke up really groggy, really tired, and that was pretty much over the past, say, 10 years or so that he has some of those kind of instances. Things that he can actually talk about is more or less things that he cannot remember. When the specific things that we would talk about, series of events or situations or episodes, or even situations of looking at television series or anything that he would be interested at the time, which at the moment he still cannot remember them. And other cases of those. So a lot of the memory lapses in those situations or in those ages throughout. There's a lot of missing information in his brain, and that's kind of one of the cases of the epilepsy case of that. In the meantime, of course, we did a couple more studies, and where he then was diagnosed with autism quite late, actually. He was 7, I think, and King's did actually a good analysis of that. So much so that we had 4 consultants going into the various environments that he was living in at that stage. So visiting at the home, visiting the school, staying with the school for a few hours, and the outcome of that was actually a good report that actually kind of determined the autism spectrum that he was in. And some part of the social aspects of that, which is, of course, he's making friends. He finds it hard. Some really lack of memory in respect to all areas of studying, for instance, as an example, or any other events in terms of our life in family. But he's here. He can now talk about exactly what you feel about what the study was for you.
I mean, one of the things I enjoyed about the study was like, I think it was sort of like a machine where instead of going to sleep, they like read how much the waves travel through your brain, and you can look at images and it'll see how high and how low it get. I remember if you grind your teeth like this, the thing would go insane, completely off the charts.
So what other interests do you have in life?
I really like talking about Godzilla a lot. Favorite thing ever. Big lizards that reach fire.
And with that comes what, the Japanese culture of anime and things like that. Yeah.
Yeah.
No. Of course, during the school, he's now finishing off his last year of GCSEs, so he's year 11. He's finding really hard to actually attain information and retain the information. That could be whether because of the epilepsy part of that was in its infancy, or was the case that of the autism itself. We don't know. At the end of the day, it doesn't matter. It's who he is, and his grades are not the greatest, and he knows of that. But at the end of the time, we just have all sorts of structures in place to try and help him and guide him through every stage of trying to attain that information and retain it as much as possible. So he's got help in school. He's got or some help. It's not official because he doesn't have an EHCP plan in place for that aspect, and in terms of governmental help and funding for that aspect. But in his case, he still has some help and does have extra time to actually do exams as well. So, and that's just part of the autism aspect of that. Nothing is. Yeah, do you want to talk about anything else that you do that you like to do?
One of the things I like to do is every evening on a Monday and Wednesday, I go to an Air Cadets base where I basically understand history of RAF, learn flying skills, and do a lot of interesting things, let's say. Most people think when I go to there is like it's really like regiment based, and it's like always like everything's strict. But when I look at it, it's just really chaotic. Like uncanny amounts of chaos everywhere. Yeah.
Okay, fair enough. So, but he's now as part of the Duke of Edinburgh as well awards, and he's gonna do some tracking and orienteering. He's almost like the Scouts at the end of the day. This is basically the RAF Air Cadet. But yeah, it's something that he's starting to enjoy to do. And this is Francisco.
That's amazing. Thank you both.
That's all right.
And it's really, I guess lovely that you had such a supportive experience at King's, actually going through the diagnostic process. It's quite a can be a long slog, I think, but that's amazing, and it's great that everything's going so well with your epilepsy at the moment. Thanks for sharing everything. It's Godzilla's 80th anniversary at the moment, is it? Is it 80?
80 years of Godzilla?
I think I heard it on the radio the other day.
I think so. Is it something like that?
No,
No?
20, no, 70.
70.
70.
The first one started in 1954.
No, there you go.
Thanks for sharing the other aspects, things like memory, where I think a lot of people don't, even some seizures are a bit hidden and people may not realize you have epilepsy, but memory is another aspect that people don't necessarily realize when it's there in the background.
Yeah.
Thank you so much for that. As questions come up, if it's all right with you, we'll ask you towards the end.
Of course.
Great, thank you.
Thank you.
Okay, so next I will present Chirag, who is working with Susanna, who's also on the call, running Leap Epilepsy. And so they've been collecting data from volunteers like Francisco, looking at the co-occurrence of epilepsy in autistic people. And so he's going to talk about some of the research that's being planned, in fact. And Chirag, your slides have come up really well. So you can go ahead.
Thank you. Just give me a second to write things out. Okay. Can you see my slides and full screen?
Yeah, that's a plan.
Brilliant. One second. Right. So hi everyone, thank you so much for being here and for giving up your time to talk about our research. I'm Chirag, a pediatric doctor and a PhD candidate at King's College London, and I run the Leap Epilepsy study. So this talk today talks about the neurobiology, or the science that we think is happening behind the co-occurrence of epilepsy and autism, and about how the Leap Epilepsy study tries to tackle some of these questions. And what I'm specifically going to talk about today is this thing called the altered brain inhibition theory. It might get technical at times, but I will go slowly and try and explain all the terms as we go along. And before I go any further, I just want to say a big thank you to all the participants who've given up so much of their time. Some participants, well, most of our participants give up about 2 to 3 days of their time, including Francisco, and it's a big onus on them. So thank you so much. And yeah, I'm just going to start. So I'm going to start with some background information on the co-occurrence of autism and epilepsy. I'm going to explain what GABA inhibition in the brain is and why it might be a potential biological link between autism and epilepsy. And then I'm going to tell you about how we plan on measuring GABA inhibition in the Leap Epilepsy study. So, as many of you know, autism is associated with several co-occurring conditions, an important one being epilepsy. Epilepsy is a condition where people are predisposed to have unprovoked recurrent seizures, and the prevalence of epilepsy is about 12% overall in autistic people and under 1% in non-autistic people. The presence of intellectual disability also plays an important role. So epilepsy is more common in autistic people with intellectual disability compared to autistic people without one. And epilepsy in autistic people, as Johnny mentioned at the start, is associated with an earlier onset of seizures compared to epilepsy in non-autistic people, and actually is unfortunately the leading cause of premature death in autistic people who have a diagnosed intellectual disability co-occurring as well. And despite this, there is extremely little systematic research on people with co-occurring autism and epilepsy. So some big questions remain unanswered. So first, we don't know why autistic people are more likely to have epilepsy, making it difficult to target specific biological pathways in the brain to treat epilepsy in autistic people. So currently, autistic people with epilepsy are given the same anti-epileptic drugs as non-autistic people with epilepsy. and we don't know whether other drugs would work better for epilepsy in autistic people. Beyond measuring intelligence, we don't know which autistic people are likely to develop seizures, and this makes it hard to know if an autistic child will develop epilepsy later in life. And finally, although we can probably imagine that it's harder to have two co-occurring diagnoses than just one, we don't know the specific outcomes in people with both autism and epilepsy. So answering these questions may help us create better seizure treatments, enable prediction of future epilepsy in autistic people, and obtain a better understanding of the mental and physical health outcomes of having the two conditions. So today I'm going to focus on this big why question. So that is, what biological changes in the brain lead to this epilepsy-autism co-occurrence? Given that there's been little research on the biology of co-occurring autism and epilepsy, where do we look first? So a first assumption could be that if the two conditions co-occur at this high rate, maybe the brain changes that lead to one condition also lead to the other. After reading through some existing studies, it looks like impaired brain inhibition might be common to both autism and epilepsy and could explain the co-occurrence. Now I'm going to give some background on this. So specifically background on what brain excitation is and what brain inhibition is. So this large pink bit on the outer surface of the brain is called the cortex, and it's made up of many different types of nerve cells called neurons. These neurons form circuits, and they connect to each other and talk to each other. And about 70% of the cortex is made up of excitatory neurons. So these excitatory neurons make neurons receiving their signal more likely to fire, and they commonly use the neurotransmitter called glutamate to communicate with each other. The other 30% of neurons are inhibitory. Inhibitory neurons make neurons receiving their signal less likely to fire, and they use the neurotransmitter GABA. So this balance of excitation and inhibition in the brain is extremely important for the brain to function properly. In both autism and epilepsy, we think that this excitation-inhibition balance is moved towards more excitation. And in epilepsy, more excitation may lead to seizure activity, while in autism, more excitation might manifest as being, for example, as being hypersensitive to sensations. I'm now going to show you some evidence that more excitation in autism and epilepsy, each occurring separately in these examples, could be due to impaired GABA inhibitory processes. And by impaired GABA, I mean that the nerve cells that produce GABA cannot effectively produce inhibitory signals. I'm just going to provide some summaries, and if anyone wants more detail, the references are on the last slide. So in epilepsy, the fact that increased excitation is due to impaired GABA inhibition is quite well established. So studies have shown that there are fewer GABA neurons, molecules, and receptors in the brains of people with epilepsy. Computational models and animal models also suggest the same. And finally, drugs that increase brain GABA and therefore increase brain inhibition are extremely common and good anti-epileptic drugs. In autism, the findings have been more mixed. So there have been findings of altered GABA inhibitory function. There's also research to show no differences in GABA function in autistic compared to non-autistic people. And in rodent studies of co-occurring autism and epilepsy, the overwhelming majority of evidence suggests that there's impaired GABAergic function when in rodent models, these two conditions co-occur. So what about studies looking at this in humans? So unfortunately, there's been no study that systematically looked at GABA impairments in people with co-occurring epilepsy and autism, and compared them to people with only epilepsy and only autism. So I'm going to quickly summarize what I've said so far. So putting all the evidence together, we know that impaired GABAergic function is associated with epilepsy. It might be associated with autism. And these animal models suggest that if you have both conditions, you may have impaired GABA. So this leads me to ask the question that if within autistic people, could greater impairments in GABA inhibitory function increase the likelihood of seizures? Of course, many things may cause autism and many things may cause epilepsy. And what we're saying here is that impaired GABA function could be part of a common final pathway that contribute to having both conditions. So in our study, we hypothesized that autistic people with co-occurring epilepsy have decreased GABA inhibitory function compared to autistic people without co-occurring epilepsy. So the AIMS2 trials, LEAP epilepsy study, aims to address the question that I've just mentioned. So here we're adding participants onto the LEAP study, which started all the way back in 2013. So the multi-site LEAP study is the largest neuroimaging study of autism in the world. It's already got 736 autistic and non-autistic participants. And now we're enriching the sample with people with, I'm going to bring my pointer out, with epilepsy only, with autism, as well as with autism and epilepsy. And when we finish recruitment, we'll have 46 participants with autism, 46 participants with autism and epilepsy, and 47 participants with epilepsy only in the LEAP epilepsy sample. And we just need 4 more people in the autism and epilepsy group to meet our target numbers. As Johnny mentioned, our study looks at people from the age of 6 through to the age of 35, and Charlotte Ty's work looks at people younger than that. So between us, we're covering quite a big age range. So now, how do we measure GABA? Until now, I might have made this sound like a fairly straightforward thing, but there are many subtypes of GABA neurons, and each GABA subtype looks different, and most importantly, as you can see at the bottom of the slide, fires electrical signals very differently. And we try and address this by measuring GABA function in 4 different ways. So I'm going to move on to a slightly technical aspect of this talk, but I'll provide a non-technical summary at the end. So when our participants come in, they give us blood and saliva samples. And I see that quite a few of our participants, quite a few of people who've participated in our study are in this talk. So that's really nice to see. And hopefully you guys can, hopefully these are good-ish memories. So we use these samples to perform genetic analyses. We perform something called GABA gene set analysis and also produce something called GABA pathway-based polygenic risk scores. And with this, we look at genetic variation in GABA processes. Then our participants go into an MRI scanner, and we use an MRI technique called magnetic resonance spectroscopy imaging. And with this, we measure the concentration of GABA molecules in the part of the brain called the thalamus. That's thought to be important, especially in epilepsy and also in autism. And this measure gives us an idea of how many GABA neurotransmitters may be available for neurons to use to talk to each other. And Francisco mentioned something about brain waves being measured during a task, and this is the EEG task. So this is what we do is we do a resting state EEG recording for 2 minutes, and the EEG measures the brain's electrical activity. And we use a measure called 1/F. That's introduced by scientists called Gao and his colleagues to estimate the average to measure the average inhibitory activity of the cortex. And this 1/F measure actually correlates with the number of GABA synapses and the firing rates of GABA neurons. And finally, our participants do a task to see how good they are at detecting vibration on their fingertips, using this device that produces vibrations on these 2 little probes that you see in the image. And how easily autistic people can detect vibration on their fingers is associated with GABA signaling pathways, and it correlates with the amount of GABA in the sensory part of your brain. So just to summarize all of that in a slightly more simple way, if we take a step back and look at all of this together, in our study, we aim to estimate GABA function in 4 different ways. So we measure GABA inhibition right from genetic predisposition to GABA molecules in the brain, to the averaged activity of GABA neurons in the cortex, and then finally perception, sensory perception that's thought to be influenced by GABA. So what might be some of the implications of this work? So if we find that altered GABA function is important in the co-occurrence of autism and epilepsy, here's what researchers could do next. So researchers could investigate if anti-epileptic drugs that target GABA are more effective in autistic people versus drugs that don't target GABA. And researchers could also measure GABA function in autistic children or younger autistic people to see if autistic children with greater alterations in GABA inhibition develop epilepsy in the future. This way, we might be able to identify autistic people who are at a higher likelihood of getting future epilepsy earlier on in the process. So this is summarized here. And the other unanswered questions that I mentioned at the start will be investigated in an epidemiology study we've recently won grant funding for. So just to summarize all of that, impaired GABA inhibition could explain the co-occurrence of epilepsy and autism. There are many types of GABA processes, and we measure GABA inhibition in 4 different ways. And our findings may improve the treatment of epilepsy in autistic people and help predict if an autistic person may or may not develop epilepsy in the future. So this is my favorite slide. So I just want to say a big thank you to so many people who have been involved in this process. So especially the participants who've given up their time and their families, who've given up their time to help us study. And the Leap Epilepsy study team, who's worked so hard to get all of this, to obtain all of this data, Susanna and Mehra, and our consultants and epilepsy consultants in several hospitals who've really given up a lot of their time. So thank you. And those are my details and references at the back for anyone who wants to see.
That's amazing. Thank you, Chirag. That was really clear. And kind of pushes towards what Chirag has been doing, which is trying to understand what the mechanism is behind people having co-occurring autism and epilepsy. He referred to this as well. So in the CANDI consortium, it's working between the ages of, I think, about 2 and 6 years old. LEAP starts at 6, which is the main AIMS study, and both of them are looking at autism. Looking at epilepsy. There is a younger study, BIBS, which looks at neonates, and so that leaves quite a gap, which is where Charlotte comes in, where she's been working with infants who have early onset epilepsy, as well as people who have a genetic cause of their epilepsy. And so I'll let her talk now. But the one thing I would say is this is a really challenging study to run. So Charlotte, since her PhD, she's a senior lecturer now, has been going around the country collecting some of this data and organizing it so people are able to, if they have a very young infant, take part in research without having to travel across the country. Okay, Charlotte, please go ahead.
Thank you. Sorry, I'm trying to find my slides. Here we go. Can you see that okay?
Yeah, it comes up great.
Lovely. Yeah, thank you very much for inviting me to be part of this webinar. It's a pleasure to be here and really interesting to hear from Chirag and about Francisco's experience as well. As Johnny said, my presentation will be focused on what the early years can tell us about the overlap between epilepsy and autism. So I wanted to first go into some background and the rationale of why we're particularly interested in early life epilepsy in the context of understanding different behavioural and developmental outcomes. And to set the scene for this, I wanted to share some of our recent engagement work with caregivers of children with early onset epilepsy. So we conducted a range of activities with these parents, including a focus group which led to this co-produced parent-voiced video. And I'm just going to show you some snippets from this to highlight some of the impact of early onset epilepsy.
Just this chronic like exhaustion all the time. Sometimes when you don't even feel that tired, you just don't feel like normal. It is overwhelming, and it is a huge responsibility that I don't think others can necessarily really appreciate. A kind of a better understanding or a more detailed understanding of likely outcomes, and that perhaps that the outcomes can be a bit varied and a bit complicated.
And it's just looking at it at different levels of needs, and it's going to depend where you and your family are, what you feel is important. So as long as all of those voices are heard and all of those things are addressed as far as possible, as long as we're listening to all of those voices, then you're doing the job right.
So I wanted to share this firstly to show how important it is that we engage with the community when we're designing research. This work has directly informed our study protocols, but also to highlight what a few of the parents were saying about varied outcomes and trajectories. And one of these is, of course, being diagnosed with autism. So the overlap between epilepsy and autism has been documented for several decades, but is becoming studied more recently. And we now know that up to 40% of individuals living with epilepsy will receive a diagnosis of or meet criteria for a diagnosis of autism. We also know, as Francisco experienced, that diagnosis of autism for individuals with epilepsy often happens a bit later, and this could possibly be due to clinical overshadowing because of a focus on managing the seizures themselves, and also separation of clinical services. But we also know that having a diagnosis of epilepsy and autism is associated with other physical and mental health difficulties, such as sleep problems and anxiety. People living with both conditions have reduced quality of life, and as Chirag mentioned, they have been associated with a shortened life expectancy. So it's really important that we try and understand what the factors are that underlie a co-diagnosis of epilepsy and autism. And doing so might tell us whether there are any mechanisms or factors involving the brain function and development that might be shared between epilepsy and autism. And this really picks up with what Chirag was talking about in his presentation. There has been some work looking at whether there are any particular factors that are associated with a co-diagnosis of epilepsy and autism. And this meta-analysis suggested that having intellectual disability, having an earlier age of seizure onset, certain types of epilepsy, including infantile spasms and focal seizures, and also the type or the cause or etiology of epilepsy seem to be quite key in understanding this. So one etiology or cause of epilepsy that we've been studying is tuberous sclerosis complex, or TSC. This is a rare genetic condition with a prevalence of around 1 in 6 to 10,000, and it's usually caused by a mutation on either the TSC1 or the TSC2 genes. And these are involved in what's called the mTOR pathway, and typically the mTOR pathway inhibits or stops cell growth. When these mutations occur, that function goes a bit wrong. So it ultimately leads to the growth of tumors or hamartomas in multiple organ systems, including the kidneys, the skin, and the brain. And the growths in the brain are associated with an increased likelihood of epilepsy. The majority of people with TSC will have a history of epilepsy, around 90%. In TSC, there are so-called second hit mutations, which ultimately lead to random variation in the number, the location, and the size of these growths. And this then leads to variation in outcomes for different individuals. In TSC, around 50% of individuals will be diagnosed with autism, but there are 50% that won't be diagnosed with autism, despite having the same genetic mutation. So this variability is interesting and might provide us with a way to track different pathways to different outcomes. Our work has suggested that early seizure severity in the first 2 years of life is particularly key for understanding later developmental outcomes. So for example, we found that the severity of infantile spasms that I mentioned predicts autistic behaviours in middle childhood and adolescence. I've put here some of the references from that work. But what's actually happening in early development? The majority of studies have been small scale and quite low level phenotyping of individuals. We know that the first few years of life are a period of rapid and complex brain development that lays the foundation for later outcomes into childhood and beyond. In TSC, the genetic mutation occurs at conception and is already going to be having cascading effects on brain development and function. Seizures that start within the first 2 or so years of life will also be impacting upon the trajectory that different children are on. But the majority of studies until recently have asked parents to try and remember what their child was like in those early years. By tracking development prospectively from birth or in the first few months of life, we can start to identify if there are any factors in early development that might predict later outcomes. And if there are, this might help us to design and target support depending on outcome. This sort of design has been used in other populations. For example, the Bastos and STARS study that you might be aware of, which is following infant siblings of older children diagnosed with autism. So we are teamed up with the STARS study to initiate the early development in tuberous sclerosis or EDIT study. And we have been following infants in this first phase from 3 months of age up to 24 months of age in a rather intensive design over 7 time points. We use the same protocol as the STARS study, and this means that we can look at whether there are any similar or different pathways to different outcomes in TSC versus infants that have this elevated familial likelihood of being diagnosed with autism. We conducted the majority of our assessments in the family home, which means we can reduce burden on families who already are traveling a lot for medical appointments and so on, and collected a range of assessments, including standardized cognitive assessment and behavioral assessment, as well as neurocognitive, more objective measures such as eye tracking. And we invited some of the infants into the baby lab to collect EEG, as Chirag talked about earlier. We recruited infants with TSC through a UK-wide network of specialist clinics and asked infants and their parents to take part in a minimum of 3 consecutive visits, but the majority of families were willing to take part in more. And we also recruited typically developing infants without a diagnosis of TSC who are age matched to the infants with TSC. And this table down here shows our numbers. We have 32 infants with TSC and 34 typically developing infants. Around 2/3 of the infants were enrolled prior to 6 months of age. So we have really rich data from the first year of life. And as you can see here, around 70% of the infants with TSC had a history of epilepsy at the 24-month time point. So I'd like to now share some of our emerging findings from the EDIT study. This is work from my brilliant student, Natasha Lindsay, that was published last year. And she's been looking at in-depth assessment of developmental ability over the first 2 years of life. So we used a measure called the Mellon Scales of Early Learning, which is a standardized cognitive assessment. And on the axis down here, I can't find my pointer, sorry. Is it this one? Sorry. We looked at the age that I just mentioned, all the different age points. And what we found is that from about 10 months of age, these 2 groups start to diverge, where the infants with TSC are showing reduced cognitive ability. And Natasha also found, sorry, I've frozen. There we go. That this slope, this change in cognitive ability was associated with epilepsy severity in the first year of life. So this supports our previous work, suggesting how important epilepsy severity is in predicting outcomes. And this is just in those first 2 years of life. And I'd now like to highlight some of our eye tracking work. So eye tracking It assesses where a baby is looking on the screen using infrared cameras. We were able to administer this in the family home. We used this pop-up tent to minimize distractions and have some standardization across different home settings. And we used a task called the gap overlap task that some of you might be familiar with, in which the baby is shown a central stimulus followed by a peripheral stimulus on the side. And so they're shifting their eye gaze from the central stimulus to the peripheral stimulus. And we looked at two conditions here, the baseline where the central stimulus disappears before the peripheral stimulus, and the overlap where the central stimulus stays on the screen, like this one, as the peripheral stimulus appears. And this overlap condition is a bit more tricky for a baby to shift their attention over to the side. And what we found here is in the infants that were typically developing, we saw what we would expect, that there was a reduction in time taken to shift their gaze over developmental time. So from 3 months to 18 months, the infants that were typically developing got quicker at shifting their attention. In TSC, from around 8 to 10 months, this reduction starts to plateau. And we found this change over time was also predicting socialization skills at 2 years of age in the TSC group. And these findings are really similar to what's been found in infants with elevated familial likelihood from the STARS study that I mentioned earlier, which might suggest that there are some similar pathways between these two groups. Of course, it's really important that we continue to follow up these children beyond 2 years of age. And we've just completed our preschool study of this cohort aged between 3 to 5 years, where we collected an in-depth assessment of different outcomes, as well as looking at things like executive function, which includes memory and school readiness. And we managed to see the majority of families, again, that were enrolled in our infant phase. And we're now planning the next phase of the study into primary school, where we want to capture more information on educational outcomes and academic attainment. We are also launching this new study called the Brain Development in Early Epilepsy Study. We're now in the thick of data collection for this. This is a really similar design to our EDIT study, but we are recruiting infants with early onset epilepsy, so it's not specific to TSC. And in this, we are asking infants and their families to take part in 4 visits. So around the time of diagnosis, where it's entirely remote, we collect online questionnaires, home video, and clinical records. And then we go to see families in their home again at 10, 14, and 24 months of age. And this time, we are collecting EEG data in the family home using wireless technology. And this will tell us a bit more about brain development and function at a much more detailed level. So we'll be able to look at brain development as a potential predictor of later outcomes. And we found data quality is acceptable from 10 months of age in the family home using this system. So what does this all mean? This really is a snapshot of the research that we're doing at the moment. But we're really aiming to find out what aspects of brain development and cognitive development predict different variable outcomes, and when we see these differences start to emerge. And by following children over many years, we can see where positive outcomes arise, and this might help us to start to design and target more informed support, and therefore addressing the priorities of caregivers in the community. By using more objective measures, so things like eye tracking and EEG, we can use these to see how effective support is in the very early years before we're able to reliably measure behaviour. So I'd now like to just thank our participants, without whom this research is not possible. As you will have gathered, it's a fairly intensive protocol, so I'm very grateful for all their time and engagement in our study. And thank you to all of the researchers and collaborators that have been involved in this work, and to our funders. And thank you very much for listening.